Hello, my name is Alyshena Case, and this is my presentation on perceptions on gene drives at Meredith College. And this project was completed in collaboration with Dr. Megan Sear in the biology department. Before I get too in-depth on my project of what I did, I should explain what is a gene drive. So a gene drive is a gene that is preferentially inherited. Some happen at near 100% ratios, and these can be either naturally occurring or synthetically modified gene drives. The picture on screen is an example of a sex biasing gene drive that is used in house mice. And as you can tell from this image, the goal of this gene drive is to get the final population to be completely male. I also should explain what gene drive technology is, since that's kind of more what was aimed on our survey. And gene drive technology is where scientists will be able to manipulate certain genes within a population to affect the probability of that gene being given to future offspring. Now that we've gotten that out of the way, I can go into a little bit more detail about my project and what was done. So here's some background information. The first thing that was done was a literature review, and the purpose of this was to gauge how previous work on gene drives perceptions was measured. This was actually mostly done by my research partner, Megan Elliott. She has not helped me this semester, but she was a big part of the beginning stages. And what she did was she did a meta-analysis of 27 articles, and we kind of decided if they were quantitative or qualitative. So quantitative articles included things that gauged perceptions numerically, such as through surveys, and then qualitative was pretty much everything else, so gauged perceptions through focus groups, breakout sessions, and individual opinions. As you can tell by the graph at the bottom of this slide, the majority of the articles that were read did contain quantitative data. After this literature review was done, Megan and I formulated our own survey questions based off of this previous literature. So we would use the questions that were in previous literature to try to figure out how we wanted to frame our survey. We did not use the same questions, but it provided a really good foundation of like how to set up a survey. After our survey was made, we then sought out institutional review board approval. And you have to do this anytime you work with human subjects in order to ensure that you're being ethical. And so we officially got IRB approval number 2012 at the end of September. After we got our IRB approval, we then launched the survey. It was online, we used Google Forms, and this survey went out at the end of September until the end of October of 2023. And we basically aim to assess primarily undergraduate student familiarity and perceptions of gene drive technology. Basically, what this means is we wanted to know what was currently known about gene drives within our community and also determine what the overall perception was of gene drive technology, so whether it was positive or negative. So now I'm going to tell you a little bit about how the survey was set up. It had 4 sections. The first section was informed consent. This was to make sure that participants knew what to expect from the survey, and you need to include this anytime you work with human subjects. The next part was the demographics section, which basically included background information and qualifiers such as gender identity, age, major, and academic status. After this was completed, they would move on to the perceptions of gene drives part of the survey, which was where we had 14 Likert survey questions and 1 yes or no question. So a Likert scale is basically a rating system that's used to measure opinions, attitudes, or behaviors. Our Likert scale was a range of 1 to 5, where 1 was strongly disagree and 5 was strongly agree. There was also one question that used this scale but had different factors. So 1 was no knowledge and 5 was expert, but it's basically an easy way to rate something. And then our 1 yes or no question was basically on if participants had heard of CRISPR used as biotechnology. After they completed this perceptions part of the survey, they got to go to the final section, which was a debrief. We offered them a debrief question, which basically allowed them to leave any input they had for us. After the data was collected, we then had to analyze the data. Thankfully, this was done by Dr. Perez in our biology department. She helped figure this out. With that, what she did was she completed 2 sample student t-tests, and we used piecewise comparisons to analyze what they meant, and I'll explain that here in a second in the results. But basically, we did have point estimates with 95% confidence interval. We did two samples. We did one that compared the mean attitude scores between STEM and non-STEM majors, and then also one on academic status, so whether individuals were in their early career at Meredith or a later career. And we defined early career as individuals that were first or second year students, and later career was third, fourth, and post-bac students. Before I get into the results, though, I wanted to show you guys what the survey questions look like. So you can see here what the participants got to see. And I'm going to give you a few seconds to kind of read it for yourselves and better understand it. All right, so here are some of the results. So these results here were taken directly from the Google survey's raw data. And basically, overall, this first graph up on the top left shows that there was overall little knowledge about gene drives on our campus. The one next to that on the right side shows that students do want this information to be more accessible. And both of these did have like a specific perception. However, the bottom response is neutral, and that kind of is included here because the neutral category is where the majority of our responses fell from the survey. However, we wanted to analyze the data a little bit more than this just to see if we could figure out any correlations or anything like that. And so that's where the data analysis that Dr. Perez did comes in. So basically, the first thing we looked at was early versus later career. And we wanted to see if there was a difference in the perceptions based off of this. So that is this graph here. And basically how this works is you can see that the difference in the mean of the responses is the little black dot on the line. The entire line is the confidence interval, which is 95%, meaning most of the responses fell within this range. If it was a negative score, that meant that there was more favor for the question from later career students. And then if there's a positive score, that meant there was more in favor from early career students. And if there's any overlap with the 0, which is the red dotted line, that means that there is no significance. And so as you can see from this graph here, all of our lines for all of our questions did cross 0, meaning that there was no statistical significance between early and later career in regards to perceptions on gene drive technology. So we repeated this process again with STEM versus non-STEM students. Same kind of idea. If it's negative, that meant STEM was more favorable. If it's positive, that meant non-STEM was more favorable. And if there's any overlap with 0, that means that there's not statistical significance. And with this, we actually did find two results that were statistically significant. So the first one you can see kind of towards the top is the opinion control mice. So STEM students were actually more in favorable of using gene drive technology on vertebrates or for controlling invasive mice populations. And then down towards the bottom, you can see attitude teach in schools was also more favorable on the STEM side, meaning that STEM students were more in favor of having this technology taught in school. So STEM students want to learn more about STEM. There were some other results, such as no moral concerns and knowledge, that was a little bit closer on the STEM side. However, it does still overlap with that 0 mark, so they are not considered statistically significant. So now, conclusions. What does this tell us? Our overall conclusion is that people simply want to know about gene drives, but they currently don't have much knowledge. And that's why we assume that the vast majority of our responses were in this neutral category. It's because people don't feel comfortable answering about something that they don't know much about. Another conclusion that we found is that STEM majors were also more likely to be familiar with clustered, regularly interspaced short palindromic repeats or CRISPR technology. And this is kind of showing that that knowledge kind of comes into play a little bit in regards to perception. And then this graph here just shows that overall, the majority of people that responded to our survey did not know about CRISPR. And then within that 46% that did know about CRISPR, it was more likely to be STEM students. So where do we go next? We're hoping to expand the survey to different institutions that have differing demographics, such as co-ed universities, because we want to determine if there's gender bias. The majority of our demographics were primarily female identifying students. So we would be interested to see if we had more male identifying students, if that would affect the data at all. And then the next thing we want to do is we want to figure out a better strategy to explain what gene drive technology is while also avoiding response bias by skewing participants' baseline knowledge or perceptions. Because in our survey, they only got one sentence explaining to them what gene drive technology is. And so we think there may be a way to give them more information, but we don't want to give them so much that it completely affects how they feel about it. So it's trying to find that equal balance that is kind of our goal to figure out for next time. And so with that, that's the end of my presentation. That's everything I found. I do want to take a few seconds to just acknowledge some people that helped along the way. Obviously, my advisor, Dr. Sear, for all of her support, Dr. Perez for running that data analysis for us and helping us with the statistics side, Austin for sponsoring me in this project, Megan Elliott for being my partner at the beginning of this presentation, or not the beginning of this project. Janie McMillan was in charge of helping us get our IRB approval, so thankful for that. Thankful for our survey participants for taking the survey and for being able to get 117 people in a month. Thankful to undergraduate research for once again funding this project and helping this opportunity happen. And then last but certainly not least is our biology department faculty and psychology department faculty and any faculty that helped send out the survey so we could get responses. With that, I would take any questions that you guys may have. Obviously on YouTube, can't really get questions, but leave them in the comments if you have any. And here's my sources. But thank you so much for all of your time.